మీ స్క్రీన్ అయితే విజిబుల్ గానే ఉంది సార్.
There's something wrong with the entire display of Zoom controls and everything. They are split into Dr. Parimi, I think we will wait for another 5-10 minutes. Is that all right?
Yes, sir. Sure, sir.
Yeah. Because
my audio is clear to you, right, sir?
Pardon?
My audio is kind of clear to you.
Very, very clear.
Okay.
Absolutely clear.
Okay.
Sir, who is sharing slides? Parimi or you are sharing?
Parimi himself.
I am only doing it.
Actually,
let him share. Let him just share. We can see.
He will do it. No. Let us wait for.
Okay.
Yeah, because sharing is permission for sharing is already given.
Oh,
yes.
No problem.
Dr. Sridhar, good morning.
Good morning, sir. Good morning.
Dr. Sridhar, yeah.
Good morning, sir. Good morning, Dr. Parimi.
There are some.
Good morning, sir. Hello, Sridhar.
Dr. Varaprasad joined. Welcome, sir. Dr. Varaprasad.
Good morning to all of you.
Good morning.
Sridhar and
Dr. Tathineni is there.
Sir, good morning, sir. Good morning, Varaprasad.
Good morning. Good morning, Ramesh garu.
Nice to see you, sir.
Lakshmi Saujanya, Dr. Chiranjeevi is there. Dr. PVR Reddy.
Yeah.
Dr. Sagar joined. Dr. Varghese. Dr. Varghese, welcome, sir. Dr. Anil Jakar.
DVR garu unnara? DVR garu unnara?
Pampichar link gaane and I think he is not able to participate in many meetings.
Right.
But I always send invitation to him.
Sure.
And Dr. Lakshmi Saujanya, Dr. Girish joined, Ms. Shahnaz, Sham Supreet. We will start in another 5 minutes.
Yes, sir. No problem.
Dr. Ratnakumari, Bhimineni. Dr. Ratnakumari, where are you from?
Sir, I'm from Agricultural College, Bapatla, sir. Good morning, sir.
Okay, I recall Ratnakumari, yes.
Sir.
How are you, ma'am?
Sir. Sir, fine, sir. Bonan, sir.
Teaching already, sir?
Sir, college, sir.
You are teaching now, okay.
Teaching, sir.
Right, good. What course is this?
Sir, 131, sir. 331, PAC 373, sir. Electric 334, sir.
Okay, good. 131 is a very good course.
Yes, sir.
Hello, Ramesh Babu. Varghese from Bangalore.
Sir?
Dr.
Abraham Varghese from Bangalore.
How are you, sir? Good morning. How are you, sir?
Good morning. How are you? How are you doing?
I am fine, sir. Thank you, sir.
Excellent.
Unable to see you, sir.
I'm not able to see me.
Okay. Now I could see, sir. Yes, I could see. Yes.
No issue. Varaprasad, good morning.
Good morning, sir. Good morning, Varghese sir.
How are you?
I am fine, thank you.
Excellent.
I am very fine, sir.
Seed park and all.
I am listening to your
seed is closed.
Sir.
New chief minister, new projects.
Oh,
new chief minister, new projects. Old one is closed.
Okay.
It is not progressing. Yeah.
Okay.
I am with Apari, sir.
Okay.
Dr. Varghese.
Yes.
Dr. Ramesh Babu is now associated with Vignan University. Dr. Ramesh Babu.
Oh, very good.
Dean, Vignan University.
Oh, excellent.
He's doing very well there.
Yes, he has always been very good.
Welcome, Dr. Shekhar, Dr. Sunil.
Good morning, sir.
Very good morning, sir.
Good.
Fine, sir. Thank you.
Yeah.
Chandrasekhar, Bonara.
Bonan, sir. Hello, sir.
Yeah, Bonan. So you are still continuing in maize. Okay, good. Always.
Sir, till retirement maize only, sir.
Yeah, but whatever it is, throughout career, you continued in one crop. That is always.
Yes, one crop, sir, one crop.
Dr. M. V. Rao garu eppudu antundevaru, rolling stone never gathers mass ani.
Dr. Srinivas, where are your colleagues from Bayers or Crop Science?
They'll join when they join, sir.
Find some time and log on to it, yeah?
Yes, sir. I know there will be few from industry, definitely.
Yeah, I know. Yeah.
Okay.
It's okay. Those the subject is such. Only those people who are actually involved, and those, of course, very seriously, people only,
Correct.
you know, come on and listen to this presentation.
I came only for the subject, you know.
Yeah. There is a limitation.
Now, is it going to be in English or Telugu?
Sir,
Which one,
it's
sir?
going to be definitely in English.
Oh, okay.
Who is that asking, Dr. Varghese?
Varghese garu. Varghese garu, this is Plant Protection Association of India, sir, not Andhra Pradesh.
ಇಲ್ಲ తెలుగులో చెప్పండి, నేను వింటా ఉన్నాను, అర్థమవుతుంది.
Varghese garu, మీరు తెలుగులో వింటారనుకు. There are wonderful people from other states also, Dr. Jakar and Dr. Desh Bandhu, Dr. there are several people, Dr. B.B. Singh, from other parts of the country also. They cannot understand Telugu. Dr. Varghese is good enough to understand.
No, in lighter vein, before that talk, in lighter vein.
Dr. Abhishek Shukla. Good morning, Dr. Shukla.
Good morning, sir.
Yeah, very good morning. So it is just 11 past 3 minutes.
Yeah.
We will wait for 2 more minutes. There are almost 50 people on the logged on to this, and there are, I am sure there are many people who joined in groups. I request all of you to keep yourself in mute. I request all of you.
Sir, this is GMV Prasad. Good morning to one and all, sir.
GMV, good morning.
Good morning, sir. Good morning. How are you, sir?
GMV, good
I'm
morning.
fine, thank you.
Sir, good morning, sir. Ramesh Babu, sir.
Good morning,
Sir,
GMV.
good morning, sir. Good morning, Sarath Babu, sir. Good morning, Srinivas.
Sir. Very good morning.
Good morning, Varaprasad.
How are you, GMV Prasad? GMV, how are you?
I'm not able to hear it.
Start. Recording started, was that? Record వచ్చేస్తే బెటర్ కదా. ఇక్కడ ఉన్నారు కంట్రోలర్ ఇక్కడ. Okay. So I think we will now start our proceedings. Very good morning, all of you. Dr. Varaprasad, there are some very good, wonderful people today in our virtual talk. Dr. Varaprasad, I welcome Dr. Varaprasad. I welcome Dr. Varghese, Dr. Ramesh Babu Tataneni, Dr. Sridhar Uppalapati, Dr. Anil Jakar, Dr. Desh Bandhu Ahuja. Wonderful to look at you, Dr. Ahuja.
Thank you.
And I look at Jeevan, Professor Kalvakuntla, Jeevan Rao. Welcome, sir. Dr. Parameswari, Dr. Kodan Ram Reddy, Dr. Sagar, Dr. Sanjeevi.
Thank you, sir. Thank you.
Yeah, I welcome each one of you. Today we have Dr. Srinivas Parimi with us. We have been conducting a series of important virtual talks. This is in the context of Plant Protection Association was established in December 1972. We are just completing 50 years of our existence, and we have planned a series of lectures connected with plant protection. And now this is the sixth talk by Dr. Srinivas Parimi. Dr. Srinivas Parimi is working as the Regional Resistant Management Lead for Bayer Crop Science, Singapore, and is recognized as a Bayer Science Fellow. His current job role entails driving resistance management strategies for durability of insecticide, herbicide, fungicide, and products with transgenic traits. Prior to this role, Dr. Srinivas worked as IRM lead, Asia Africa Hub in Monsanto, and as group leader entomology in Maiko, India. Srinivas obtained his PhD in entomology from University of Nebraska, Lincoln, USA, and worked as postdoctoral research associate in insecticide resistant management at the same university. He obtained his bachelor's and master's in agriculture from Andhra Pradesh Agricultural University and worked as a research associate at ICISAT, India. Srinivas has been working in the APAC region for the past 2 decades in crop improvement, native resistance breeding, trait evaluation, gene discovery support, regulatory stewardship, regulatory advocacy, and resistant management in more than six crops. He has 27 peer-reviewed publications, co-authored 3 book chapters besides several oral and invited presentations across globe. He is the current chair of the Asia IRAC Insecticide Resistance Action Committee, member of the IRAC International Crop Protection Working Group, and represents the industry as a technical expert on fall armyworm in this region. So this is in brief about, he did not want me to, do a lot of, about his personality and all. So very briefly, he asked me to talk about his, his professional association as a resistance entomologist. In fact, Dr. Parimi was also, part of our executive plant protection association in the past. So Dr. Parimi is not a new entry to Plant Protection Association of India. We have worked with him, and he was as executive member of Plant Protection Association of India. He served the society for almost 3 years. So without wasting much time, in fact, I would request you all to keep yourself in mute yourself and listen to him carefully. And I think screen sharing has already been given to Dr. Parimi. I welcome Dr. Parimi to start his presentation, and the floor is yours, Dr. Parimi.
Thank you, sir. Let me first share my screen.
Baba, I share the pieces of the sand. I sing to the that checker. I do it in the
Please put it.
start to make
You can host can mute all. Host can mute all.
Just, okay. Just thank you, sir. I hope you all can see my laptop screen that I'm sharing now. Yeah. So
Yes.
thank you so much for the introduction, sir. Rightly said, I have been associated with the Plant Protection Association of India for a long time now. In fact, I'm a life member. Thanks to Surinder Garu, once upon a time, he encouraged me to do that. Having said that, I am thankful to Dr. Sarath Babu, who invited me to talk something that is close to our heart. Most of the entomologists, they like to hear about pest management. And before I start talking about what I'm going to present today, I would really like to acknowledge the presence of all senior colleagues, my professors, Dr. Ramesh Babu, Director Dr. Varghese, and many others whom I may have met, Dr. Varaprasad. I have met them briefly. And I'm also seeing my classmates, few of them here. And I also am seeing some industry colleagues. So, which is very nice. It's a nice blend of audience for me after a long, long time to talk and share my thoughts about pest management and resistance management. And thank you for your presence here. And I appreciate it in my heart. Having said that, the title seems very flashy. It says Confluence of Modern Day Pest Management and Resistance Management. And what I'll do is I'll try to take you through a journey of what we have been doing in pest management and how things have evolved over time in pest management. Also related to resistance management and why it is so important today. Although both these concepts have been there since maybe 5-6 decades around us. So this is a very standard slide. We all know food security is a major issue for all of us. And in countries like India and Asian region, it needs there's a lot of food demand in the next few decades. And most of these issues are, surrounded between, the demand and supply and making these foods. Of course, there's always a gap and reasons are many between demand and supply, why there is a gap. And also there are certain core crops or focus crops where this demand and supply gaps are the maximum. I think you're also seeing this. Let me minimize this. Having said that, the important thing is that the productivity. If you look at the fields in India versus other parts of the world, for some reason or other, which we all are aware, or a combination of them, we tend to be seeing lower yields in India. And it's, like I said, it's a combination of reasons. Adding to these macro challenges, we have all of these challenges at the farm level. You name it, insects, diseases, environmental stress, nutrient deficiencies. And in the recent times, you started seeing a lot of viral diseases emanating in vegetable crops, particularly. So all of these, they pose the challenges to us as scientists, for sure. But more importantly, if you really look at it at a practical level, it's a challenge for the farmer. Just imagine how confusing it is for him to manage a leaf feeder or a borer and suddenly a sucking pest coming at the same time. So that's where, as scientists, we can contribute better with our research programs, whether it's public or private industry. Adding to all of this is the unexpected, the invasive species. Yeah. So, yeah, I'm hearing background sounds still continuously. Yeah. So, thank you. So, these challenges. We in India, since almost mid 2015 or 14 onwards, we are facing these challenges. It can be Tuta absoluta that came in around early 2010 and 2014 in between, or Thrips parvispinus, which is recently causing havoc in areas around Guntur and chili growing regions of the country, and of course Fallout. I'm only naming a handful of them, but if you really look at it, these are surprise things for a farmer. He would never be able to withstand these surprise challenges, even scientists. So for all of this, we go back to our classical word of IPM. It's easy to say IPM, integrated pest management. You recommend things. All of us put our brains together. We come up with a list of things. But just imagine on the farmer's side, what does he want? He wants some immediate results. And that's where the challenge comes. In any pest management paradigm, IPM paradigm, the base of it is provided by what you call this LO region, the actual native resistance, linked to how we understand the pest biology, ecology, and if there are any biological and cultural methods that you can follow. But if one of them most importantly, when you don't know the pest biology and ecology, you can't do much. That's a given. Once you understand all of this, then you start detecting the presence, monitoring it, and the final step is to go with insecticide. At the end of the day, we all are looking at yield. Does IPM really affect yield or yield more? In fact, it doesn't. So when the messaging from any office is going out, it should be very clear that we are only trying to protect the yield by creating the balance in implementing some of these measures and targeting the pests that are of importance for us in the field. Overall, IPM is a very conceptually very strong concept if you want to implement it. But there are several challenges to it. And I don't have to explain in detail, but the biggest challenges are the limitations around some of the biological control agents, availability of them, supply of them, efficacy of some of the insecticides that we talk about, which farmers needs to understand properly. Native resistance. It's one of those areas which is very loosely used, but again, we don't want to explore any further after a certain level. Native resistance to lepidopteran pest, in my opinion, is a myth. It is almost outdated to talk about native resistance to lepidopteran pest. However, you will get some responses in certain crops which are very specific. Whereas native resistance to sucking pests is so well established in our system, we need to exploit more. But are we doing enough is the question mark. So at the end of this entire management of a pest in a field, what farmers do? They resort to the easiest approach, use of insecticides. And when they go for use of these insecticides, particularly same insecticides, similar insecticides, it can become a problem. Why is that? When I'm saying similar or same, I'll explain in the following slides what I am referring to. But over-reliance or use of insecticides is not a problem. It's over-reliance on the same and similar insecticides.
No.
In general, insecticide mutations, resistant mutations that are in, we call them alleles, are present. So yeah, so these mutations, what happens in the real field scenario is when you start using the same or similar mode of action insecticide, they get amplified. So they get selected. More and more insects will start, getting selected for these mutations. And at the end, you see all the greens gone and getting replaced by yellows and reds and blues. And this is the dangerous part of using insecticides again and again that belong to the same group particularly. And that's what farmers do, because they only have an option or a cheaper option, whatever you call it, with them. So is it good for us? Is it good for the farmer? Definitely. And that's where we get into this conundrum of events where we say, okay, we need to manage this resistance. Instead of managing a resistance that already happened, can we prevent it? Can we delay it? That should be the question now. So earlier, the thought process was resistance has occurred, we could not detect it. So how do we manage it now? Now the rhetoric and the question should be different. And we as industry, we already started doing it in the past few years, maybe close to a decade. How do you prevent or delay? That is the key. If you can prevent and delay it, then you are protecting a management option for the farmer to use in the long run. There are several strategies that can prevent and delay. starting from the type of application you're doing, the nozzle you're using, etc. and etc. But there are three key things that one has to be keeping in mind when you start looking at preventing or delaying resistance. One is monitoring. How do we monitor this resistance development or in the insects? The second is rotation or alternation of MOAs. I'll explain in the following slide what I am referring to as MOA. And third, the most important thing is integrate as many possibilities or strategies or measures that you can deploy in a crop pest system to manage it. These are very important. Although economic injury level and threshold levels are spoken about, it is not easy to have thresholds developed for everything, every insect in a crop system. And we as entomologists know that very well, that it's impossible to do that. So the focus should be on what are the strategies integrated way you can use, how best you can manage and monitor the pest. These are the three things. In the next few slides, I'll try to explain what I am talking about MOAs. MOA is the short form of mode of action. Industry kind of embarked on this journey through CropLife International. So there are members under CropLife International who voluntarily committed to have these mode of action icons on their product labels by 2023. And I believe this is the most critical information that all stakeholders in the supply chain of pesticides all the way up to the end customer needs to understand. And it has a guidance document developed and icons, how they need to be presented, everything is all given. And industry already shifted towards this. When I'm saying industry, these are the CropLife members companies. Not all companies do this. So this MOA labeling is a shift from the way we have been thinking, how you create awareness among farmers and how you educate farmers and contribute to resistance management and overall pest management, finally ensuring product durability. This MOA initiative is key to the success of product longevity when you look at any pesticide. However, I'll explain briefly using an insecticide example. So this is the typical label of an insecticide. And you will have all the active ingredient information here, caution, everything. So now we as CropLife member companies, we started putting this label here, or this group, insecticide and 15. What is this group? This is the group. So all insecticides are classified based on their mode of action into different groups. You can go to the website of IRAC and you will get all the information. In the earlier generation, there like 3-4 decades ago, there are only pesticides belonging to 3 groups the most, or maximum 5. Today, there are 30 groups identified with different modes of action. It's challenging for anybody to remember while recommending, while executing at the farm level. So it was felt very strongly that we start using these numbers when we go to the farmers level. As an example, we started putting this. If there is 1 insecticide, you write the group name here as 1A. 2 insecticides, you can represent like this or this. If there is 3 insecticides, sorry, an insecticide and a fungicide, you can always do this. Okay. And this is a very standard method implemented across all CropLife member companies in industry now. And similar modes of action were also developed and created, and they are part of the training material by Herbicide Resistance Action Committee and Fungicide Resistance Action Committee. And to let you know, there is also a new nematode grouping that was done also for reference purposes. Having said all of this, what's the importance of mode of action? Basically, it tells you don't use the same mode of action, which would enhance the selection of resistance. Education to the growers, extension workers, or other stakeholders will be easier. And when we say rotate MOAs, it's easy for them to know based on the number that they can use. If there are 3 very effective and efficient MOAs or insecticides available with them, they need to rotate them. So as an overall initiative for resistance management, it is very critical component now. Having said that, are all the countries following this? No. There are only a few countries in Asia Pacific where it is mandatory. Interestingly, the big countries, China, India, it's voluntary. So when it's voluntary, what it turns out is only the crop life member companies have this. And it is not part of the registration process, and nobody can really ask at a company why you are not doing it. So whereas there are other countries in Asia Pacific which made it mandatory. And that's a big step. And in the past few years that some governments were able to do it very proactively. So the key to all of this mode of action labeling is to ensure that we have this information given to the farmer to ensure proper management at the ground level. At the end, the goals of insecticide resistance management focus primarily on 3 areas. 1, to remove these resistant alleles from the population if they start seeing it. So they are inducing reduced fitness in the insect population. You need to preserve these susceptible genotypes in the population. While doing these 2, you need to ensure there is a redundant killing. I highlighted few things from what can be done to achieve these goals. And if you look at it, again, it says use different MOAs and ensure IPM practices. So this is the key, and this is the confluence of integrated pest management and insecticide resistance management. They go hand in hand. And with that is the MOA labeling that we all embarked upon, and we want to see it more and more happening at the ground level as industry. For the next few slides, I'll take you through IPM components, classical IPM components, how they have evolved over time, and how we all should be looking at in this country in using some of these components. I am not going to explain everything, but I want to highlight on 3 important components, particularly the host plant resistance or crop improvement, genetic control, what is new in it, a little bit on the pheromones and how mating confusion is being looked at. And then I'll also explain a little bit about how resistance management tools also are evolving. Our classical approach has been, particularly in the use of insecticides, to develop an insecticide, throw it on the farmer, let him use it. But at least for us in Bayer, what we are doing now is we are trying to kind of understand the needs from the market in prior, create a product concept, and take these potential candidates for testing. At the same time, start generating technical data that supports these products over a long period of time, not as a solo product or a product concept, but also how any kind of risks that may emanate from issues such as resistance development can be mitigated as part of the life cycle management, we call it. So we are giving a product concept to the farmer. We are looking to do that more and more now than we were doing earlier. And one of the, I just want to highlight this, one of these is recently released, which is a diamide, and it's a very effective compound. It's called Viago. Why I'm showing this is not to advertise something. The life cycle management of this product is being built very strongly, and one of the major countries is India. What you're seeing in the video is actual assay that we did in the labs, and basically you see these are, I believe, Falernum larvae that are feeding and dying on the right side with Viago treatments. Not to dwell on product more, but I was talking about host plant resistance. We all know the classical host plant resistance includes constitutive resistance and induced resistance. In constitutive resistance, we know very well, we do a lot of screening, establish the mechanism, and then come up with a line or a variety or a hybrid that can be used commercially. And this is a typical example of host plant resistance done for rice BPH resistance. After you do all of this, you'll find some plants surviving, some plants don't. We rate them as resistant or tolerant, and you take it to the next level. This is for the sucking pest complex. I mentioned earlier for lepidopteran pests, it's very, very challenging to develop this kind of native resistance trait. So over a period, scientists realized that it's kind of difficult, and that's where we entered into the era of BT crops. BT crops are considered an integral part of IPM, and they're an extreme case of host plant resistance in a way which affects the pest biology, or it is called antibiosis. So earlier, we very well know the single gene products of BT crops that have been kind of sold in the market. In this country, we all know Bolgar and Bolgar 2 as one of those products, first and second generation cottons that are sold here. But if you really look now, this is the global scenario. None of these products are available in India, but they're available in Asia Pacific, they're available globally in many other regions. Smart Stacks is a 7-gene product. And it amazes me how we came this far with a product that would really manage above the ground pests, below the ground pests, offers weed resistance or herbicide tolerance. One of the key products that I want to talk about for a few seconds is BT Double Pro here. This is a cotton BT corn technology, and it has 2 genes with 2 different modes of action. I was talking about modes of actions earlier, and also offers herbicide tolerance. Why I'm bringing it up here today? In the event of Fowlermium invasion recently, in most parts of Asia, where it's grown, that is Vietnam and Philippines to be specific, this is the only product probably which was like withstanding very, very well against Fowlermium infestation. And we were testing this product in India a long time ago, but we could not really bring it out into the market. So in my opinion, it's a lost opportunity. Whatever we could have done, we could have done at the right time, but now the only opportunity that we have is to look at these kinds of technologies, if not BT, the other technologies to be brought into India. The next one that I want to touch base a little bit is genome editing or gene editing. It's the fad word these days in technologies, and it's a technology that's having impact not just in agriculture, everywhere, including medicine and health. I'm not going to explain a whole lot about it, but what I want to say is this is one type of a tool that can be used to create a very site-directed insertion or a deletion or editing in a genome. Very simple. It is a little different from transgenics. It's more of a cisgenic technology in a way when you have a site-directed insertion. Lots of policies around globally. All that are in blue here, there are policy discussions happening with the governments. And nothing is clear yet how these gene-edited crops are going to be treated. The only 2 countries or 3 countries in Asia Pacific that allow, or at least they do a case-by-case determination, are Japan, Philippines, and Australia. Whereas the best news that one could hear in this area is from UK recently, that they relaxed some of their regulations in relation to gene-edited crops. There are lots of products in the pipeline. What I want to show is, look at the benefits that are actually perceived already from the research that's happening around in different areas. Agronomic benefits, shelf life of the products, nutritional benefits. And the 2 products I will be talking in the next slide. This slide is mostly talking about the research that is in one crop today. This is all rice-related research that's happening, and this was very well summarized by Zafar et al. In 2020. So the 2 products that I really want to highlight are these 2. One on the left-hand side is the one, it's a high oleic soybean oil in the US market, released by a company called Calixt. It's Calino oil. This is the first product based released or commercialized based upon gene editing technology. And the one that is closer to us that's recently approved for sale is GABA tomatoes, high GABA tomatoes. So GABA is an inhibitory excitatory neurotransmitter. So it kind of lowers your blood pressure apparently as a neurohormone. So these tomatoes express that GABA and they are on sale. This is the advertisement from their website. And it's a collaborative effort of Santec Seeds, University of Tsukuba in Japan, and it's marketed by Pioneer Ecoscience. So we are seeing these products out already, and very soon we may see these products around us. The last one in crop improvement area that I want to talk about is MAGIC. It's multi-parent advanced generation intercross. It's nothing hi-fi, no tools required, actual breeding that can be done using the elite lines, which are very well characterized. So if you have few elite lines and you want to you're trying continuously over years to kind of bring your wanted trait into your products, this is a great way of doing it. In fact, I should very proudly say I had first-hand experience in doing it in a different crop. So you select all the lines, you make these diallel, hard diallel matings, or the generations, you start selecting them based on phenotyping, which is the best way, I still believe. Of course, integrate some trait-associated marker-based selections, which would have an impact because you can speed up the season if you can in between. And at the end, you will end up with a population of MAGIC. And that population can be used for any purpose because you already have well characterized. This can become a basis for crop improvement. And we already are seeing a lot of activity in rice, cotton, wheat, soybean, and maize for different characters. And this is one of the best approaches today that can be used to incorporate multiple pest resistance if once it's very well characterized. The next couple of slides, I'll be talking about a technology called sterile insect release. We all are aware that SIT is a sterile insect release technology. And traditionally, it was like you used to do some radiation, sex separation, sterilization, and release. And it's very well established in many areas around. However, the recent advancements in the past decade or so by a company called Oxitec, based out of UK, they kind of eliminated this need for irradiation. So the genetically modified strains of what they call friendly technology, friendly insect technology, has a self-limiting gene in it. This self-limiting gene is repressed on a tetracycline or antibiotic-based diet. So in the lab stage, they're always on the antibiotic-based diet. And when they're released into the environment, basically, once they're outside of this tetracycline or antibiotic-based diet, the gene starts expressing. And it expresses so much, it overloads or overproduced and shuts down the transcriptional machinery in the insect. At the end, the high level of expression kills the insect. The biosafety of it, all the questions you can find on their website. It's very interesting to learn about it. At the end, you release it, the progeny dies, and even the released insects also eventually die. It's a technology that is very well studied now in public health area, and slowly they're testing in agriculturally important pests. You can visit their website, you'll see a lot of information. The thing that I want to highlight is three different cases. One is the first US field test of GM mosquitoes. It began after the approvals in last October or August. And the results have been shown to be very, very exciting. It is in an area called Florida Keys, which is having a lot of mosquito-related vector diseases. And I sincerely forgot the mosquito species, but definitely they have tested these mosquitoes and they found a lot of success in terms of reduction in population. Brazil is the first country to bring out a product, a groundbreaking commercial launch, they call it, of friendly ADC. And it suppressed populations very well in all the field studies conducted. And the most important, that is of kind of linking to this talk, is the recent publication in 2022, where the self-limiting falloarmyworm is seen as a tool from the same company and a research collaboration with Bayer Crop Science, that a new approach for resistance management. I mean, the two graphs in this study, what they show is basically when they're used, when the incorporation of the GM insect or GM falloarmyworm trait into the models, it shows that the longevity of the durability of the refuge deployment and reduction in the BT resistant falloarmyworms in Brazil. And the transgenic insects also within few generations disappeared from the environment. So overall, it's a great tool for resistance management and also pest management. If somebody is interested, I'm happy to share this publication, or you can search for the title, you'll easily find it. And I believe this is on Oxitec's website too. So here is a tool which you can efficiently use. And they also have a partner in India, which is called GPIT. And again, you can find those details on their website. And this is a kind of a very, very interesting thing that I found while I was preparing for this talk. Nanopore sequencing technology is very recent, probably a decade or a decade and a half old. And it's from Oxford. You can see on the left-hand side, the device in the hand of that picture. That's how big the device is. And if you notice, these are all the users all across the world looking at different drug-resistant bacteria and detection of viruses across the globe. Even in India, there are users for this. Why it is such a big deal? It is a portable sequencing device. And it's a real-time sequencing device which can read up to 1 megabytes of size of sequences. It's a proven technology. And if you visit their website, so many interesting publications to read about it. And the investment is not so huge. If you really look at the lakhs of rupees we spend on equipment in our labs, this costs you around $1,000 to $2,000, depending on the size. It's called Minion. And the users are indicated in this graph here for different purposes. But if you look at all the users of this equipment or this tiny piece, which is a little bigger size than a USB drive, you can see how valuable it is. It is recently used in Pakistan, a publication to look at SARS-CoV transmission cases. It's an excellent surveillance tool proven to be used against plant pathogens in plant and insect tissues. These are not research studies alone. The work has already started in using these at the ground level. And the bottommost one, the most important, the insecticide resistance in malaria vectors in Africa are being monitored using this tool. So it's not very far away that these kinds of tools can be used in pest management for monitoring pests. So one of the valuable inventions, if you really ask me, in this area of public health and resistance management, diagnostics, this is one of it. And they have different size instruments. This is called Minion. Okay. Lastly, I want to talk about pheromones. Pheromones, we know very well they're used for monitoring, mass trapping, mating disruption. But all of these are tried and tested with lots of limitations on the execution level. And I don't have to explain what are those limitations. But what I want to talk about is this this technology that actually lowers the production costs. And that was a limitation in producing large volumes of these pheromones. And this was a barrier for a long time, the cost production of these pheromones. And after the Nobel Prize in Chemistry in 2018, these scientists, they invented or discovered a method to produce these pheromones and increasing their yields, reducing the cost. and it's by a company based out of US. There's a lot of testing happening in Southeast Asia. I believe they even started testing in India. And why I'm talking this in the context of pest management or resistance management, if one can show the value of these pheromones blends for use as a large volumes, it's a huge thing for pest management. We all know the problems in installing pheromone traps. The farmers, somebody kind of takes them away from the fields, and the farmer is at the loss. So I believe this company also is working on sprayable methods of using these pheromone blends by producing them in huge volumes. So in few years or less than a decade, our IPM entire components can change. I didn't talk a lot about digital agriculture because drones is a big topic. I don't want to get into it. You all are aware how drones are being used left, right, and center in every area around us. But this is how the IPM components will change in the future. With that, I want to leave a couple of slides and a message to that IPM and IRM, they go hand in hand. And as Bayer, we believe in it. As a scientist, I believe in it. And it's very important that we use both the best out of the components of IPM and IRM and ensure longevity of the products. And you know the value of IPM and IRM, and it saves money, it saves time. Safety is another component that we always look at, and it enhances the safety of the produce, particularly besides farmer personal safety. So at the end, we as Bayer, we work in seeds and traits, crop protection area, and also use digital tools in supporting farmers and enhancing the yields. Our focus in the next few years to come would be around use of digital tools and reaching out to as many farmers and enlarging the footprint to reach and educate the farmers. These are the new initiatives as Bayer to the small farm holders in Asia Pacific. And India is the key region, key country for us. And more importantly, it's not just farmers. We are also interested in creating the next generation with scholarships, partnerships, and creating communities around us. And I think we already embarked on these initiatives and trying to find new space to work with younger generation. With that, I'll stop here and thank you very much for your attention. And I'll take any questions now.
Yeah, Dr. Parimi, it was a wonderful, talk. Everybody it was actually a spellbound presentation, and I'm sure not really, exaggerating the presentation, but it is really, that's the reason why I wanted industrial participation also in the series of, the virtual talks that we have initiated at Plant Protection Association of India. I really appreciate you for the new dimensions and the new initiatives that the industry has been into, especially your, multinational institution. And I am really on behalf of my entire community of scientists from public sector institutions and also from the members of the Plant Protection Association of India, really are grateful for this wonderful presentation.
Sure.
And I think some of your colleagues are also, there's one Dr. Susan Knight from, I think somewhere from abroad. Maybe she may be from Bayer Crop Science. Good to see you, Dr. Susan Knight. And now I would look forward to a little bit of interaction and expert comments from expert views from Dr. Varaprasad and Dr. Ramesh Tathineni, maybe from Dr. Varghese also. Now, let me look forward to Dr. Varaprasad. Dr. Varaprasad, please. Dr. Varaprasad, are you there? Okay. Now, Dr. Tathineni, Ramesh Babu. He was, in fact, Dr. Ramesh Babu was his guide, Srinivas guide in MSc course. I would like Dr. Dr. Ramesh Babu to come forward.
Thank you, Dr. Seth Babu.
Yeah, please.
Thanks, Dr. Srinivas. Very useful lecture. That too, in the present context, the IPM and IRM are to be hand in hand. And in the IPM itself, we have shown very important aspect of area-wide operation. That is, we have given top priority for that. Whatever the technologies we suggest I promote, but area-wide, application, if it is not there, it will go waste. So that implementation point of view, extension point of view also you have touched upon. And your magic lecture has included the magic that magic in the sense multi-parent advanced generation, intercross also you have projected. Likewise, sterile techniques which have been projected, very recent sterile techniques also you have projected. Overall, the lecture was very useful. And certainly, I think every entomologist has to take care of both IPM and INM. We cannot look both separately, and we have to see that particularly we need to integrate both the IPM and IRM. Then only we can protect the crops from the pests and also to help the farmers to reap their yields and profitable agriculture. Then with these brief remarks, I know your situation. I think it is there is nothing wrong to inform here that Dr. Srinivas' mother is critical. In spite of that, he doesn't want to lose this opportunity because so many scientists are waiting for to listen to him, and immediately after this lecture, he has to rush to hospital because his brother is looking after his mother. Of course, she was not well for the past so many months, but yesterday she was critical. Dr. Srinivas, don't worry, be brave. She will come out of the problem. And after this lecture, you can certainly go to immediately the hospital and look after your mother. And thank you very much. He was my first MSc student in 91-93 and best student also. Thank you, Srinivas, and
Wonderful.
I hope everybody got benefited.
Yeah, thank you, Dr. Ramesh Babu. Before I move on to questions, I would like to give you a few comments from the audience. Dr. Deshbandhu says, it is very informative talk, and I am very sure that the information presented here is of great use in implementing IPM program, vis-a-vis reducing that curtailing the pesticide consumption in India, particularly in horticulture system. Congratulations to Sharad Babu and his team for inviting the speaker to share his views. And Dr. YG Prasad, highly informative talk, compliments to you. And Dr. Gundappa says, very informative talk. Dr. Susan Knight says, excellent presentation, Srinivas, and thank you to the Plant Protection Association of India for the opportunity to join. Dr. Shekhar says, excellent presentation. Dr. Jagdish says, this is a nice presentation and updated new area of research for pest management. Dr. A.P. Padma Kumari says, Srinivas, nice talking, highlighting the changing paradigms of IPM. Thanks to the organizers. Also, Dr. YG Prasad says, two questions. Combination insecticides, even with different MOAs, still need farmer education. And when to use, example is Profex and cotton. Profex on cotton being used early for PBW control on BT cotton, not ideal. Do you get this question, Dr. Srinivas?
Actually, yes, and I didn't get Profex. What is Profex? Maybe it's Profenofos, if I'm not wrong.
I am also not
Dr.
sure.
Prasad?
Yeah, it is Profenofos and cypermethrin combination.
Okay.
Correct. Yeah. Correct. Yeah. So actually, combination of use of insecticides is generally recommended or used by industry to, if I may say, expand the spectrum of both the insecticides and also ensure farmers uses it. We are also looking at how best to recommend some of these mixtures in the crops. And I think we have to be careful in how these mixtures are developed. If they're from the same MOA, we don't encourage it. we don't use it. Of course, there are tank mixes happening at the farmers level. That again, we are trying to, we already have a position statement, but it doesn't help. I know that. We are trying to educate it throughout, chains, marketing chains, that how best to use these mixtures and how not. Now, coming to the issue of Profex on cotton, how ideal it is, I cannot comment, sir. But one thing for sure, I know that pink bollworm moths are highly susceptible to pyrethroids, and that's one of the reason why it is recommended at the early generation, on the early generation pink bollworm emergence. So the moths are there, so immediately you can control the moths with pyrethroids. And I think Profenofos is coming into picture because of other larval control. So that's the way I'll put it. If it is ideal, yeah, we can always challenge it. But you don't have many molecules that you can use in cotton now. And if there is something new coming, that's always useful. And I also see your question of usefulness of RIB in cotton for pink bollworm. Now, I think as a country, we are late a little bit in getting the seed blend approval and all. We did our best. It took us 5 years to get the seed blend or RIB approvals in India. Lots of conversations, negotiations, etc. So it's a compromised thing for pink bollworm control. But I'm pretty sure the RIB deployment in BT cotton is going to help for other insects that are already Bolga 2 technology is working against. Did it? Okay. I hope I answered your question, sir. Yeah.
Okay, to move on, Dr. Sridhar says, excellent information shared, Dr. Srinivas, in a lucid way. Dr. Lakshmi MSM says, very informative talk and wonderful presentation. Thank you so much. Dr. Anil Jakar says, excellent presentation. Dr. Varaprasad, excellent lecture, very thought-provoking. Congratulations, Dr. Srinivas.
Thank you, sir.
J. Sai Ram, nice and informative talk. Reji Prasad again, BG3 cotton in Australia. Do we see a need for India?
That's a excellent question, sir. Actually, BG3 in itself is the Taiwan product that I was showing you earlier. So it has a gene that actually works on ligers and thrips, and it's I think it's believed to be released in US. And in Australia, it is approved for release, or it is at the final stage for commercial sale. Either way, it has a value in India, definitely. But a bigger value, I think we are seeing now is with the herbicide tolerance trait of Roundup Ready-Flex. Definitely, there's value for both of these technologies in India.
Okay, Dr. S.D. Mahapatra says, nice presentation. Thank you. Dr. Sunil. And Sunil says, excellent presentation, giving exposure to latest array of cutting-edge technologies available in plant protection. Thanks, Dr. Srinivas. Dr. Nivrutti Sonan says, nice presentation, sir. Dr. Anil Jakar says, Dr. Parimi, I want to hear your views about the fate of combi products and insect resistance. Briefly, please. Dr.
Yes,
Srinivas.
I think I kind of answered this earlier in Prasad's question, but one thing I want to say is the product combinations which are from the same modes of or similar mode of action that I was talking about earlier in the presentation, they won't help us in any way for managing resistance or overall. But if they're offering two different modes of action against a target, then yes. But if they're used against different targets, they're only contributing to pest management. They're not contributing to resistance management. So it's how each of the industry looks at these products and how they want to bring. So, but if it is tank mixes, we can definitely suggest the farmer to use products from different MOAs and use them against a target.
Okay, let's move forward. Dr. Rajeshri says, very interesting and futuristic talk, Dr. Parimi. Dr. Padma Kumari, what's your opinion on lack of general purpose insecticides in early stage pest control in rice and the use of diamides in early season pest control in rice though its recommended only at 50 days after transplantation? Do we have some answer for this?
Ma'am, you threw a googly at me, actually. So it's general purpose. When you're saying, it's like it becomes a broad spectrum or a general purpose control, which I think is not advisable in any crop. However, in rice, if there is a need in the early stages for stem borer management, or if there is a need for BPH management, definitely yes, those recommendations stand. I am not a proponent of general purpose control of spraying anything, and of course, use of diamides is always encouraged when there is a target pest visible and crossing certain thresholds. And I think in rice particularly, these instances start from around anywhere from 35 days after transplanting, with stem borer infestations increasing. So maybe that's why the recommendation. But I have not checked the recommendation for India, ma'am. I'll be honest with you.
Okay, Dr. Rajasri has a question. Any solution for fall armyworm after entering into the maize cobs? I don't know, maybe a general question. Kindly say something. Yeah.
I'm shaking my head. No, ma'am. No. Once fall armyworm enters into the cobs, or in fact, what our recommendation from any pesticide company is that not to spray anything once the crop goes beyond waist height, your waist height. You know, that's the height of the crop that it should be. After that, it's not advised as a safe use perspective and a stewardship perspective. So we don't recommend. And there's no value in spraying the cobs anyway.
Okay. RRU entomology, RERS says good talk, Srinivas, and there is a question from their side. Are combi products good IRM strategy in the long run?
Yeah, I think I already answered.
I mean, already answered.
It depends on each of the crop and pest system, if I may say that. So I cannot say with a blanket recommendation of mixtures are good, but you have to look at each of the crop pest system. If there are other options available, I wouldn't wait. I wouldn't immediately jump into mixtures. Sequential applications are always good in these kinds of scenarios, but yeah, it's a case-by-case situation.
Dr. Sridhar asks you, based on the life cycle duration of insects, how should we plan MOA-based rotation of insecticides as per IRAC?
As per IRAC, it is what we call treatment window-based application. And a treatment window is approximately 30 days treatment window, assuming a lepidopteran pest goes through 30 days of life cycle. So that's how it is as on date for lepidopteran pests. Whereas for sucking pests, it could vary a little bit. We directly do not recommend any treatment window-based application, but I have to check if there is a different recommendation for that. I don't remember top of my head. If Pampa from Cortiva is here, maybe he can add, because he was working with the global teams to develop sucking pest related guidelines, right?
Okay, thank you, Dr. C. Is there anybody? I mean, Dr. Varaprasad, are you back? Dr. Saab?
Yes. I'm here.
Actually, I was calling for you. I think you
were. I heard it. There was some. No, it was internet issue, something. Not able to get in. In between, also, I lost a few minutes.
Your views on this excellent presentation, Dr. Varaprasad.
Oh, I have already given. Congratulations. Very well done. I think my generic concern is why it took so long time for seed treatment in the regulatory issue. So how do we really public-private partnership and how do we take it forward is some general concern. Otherwise, I am fine with excellent lecture and a good approach that given by Dr. Parimi. Thank you.
Any feedback on Dr. Varaprasad's view, Dr. Parimi?
Seed treatment, yeah, that's a good question actually. It's also, sir, to put it, when you go for a seed treatment, you need to have right molecule to do it and right formulation to do it. I think it has evolved over years after our initial attempts of using neonicotinoids as the seed treatment option. Biologically also, we never really studied so well if how early season lepidopteran control can be brought out with the available chemistries. I think this has come out very well in the past, say, decade and a half or decade only. And now you are able to see some seed treatment options for lepidopteran control. Maybe that's the delay. Otherwise, the intent has been there because you already know how gaucho is used or how other chemistries like thiamethoxam or other neonicotinoids were used. So the intent has been there all through. It is just that the availability of the right chemistry, right formulation technology, choosing the right.
Anything urgent?
Dr. Srinivas Babu, can I have a couple of queries? Dr. Srinivas?
Who is it?
I am Dr. Rahman.
Oh, Dr. Rahman.
Sir.
Yeah, I am fine, sir.
Dr. Rahman, hold on. There is one last comment. I just want to finish this.
Sure.
There is one SV Gopala Swami. He's asking excellent session, sir. Why store pest management is neglected by industry? Any new thing to offer?
Dr. Gopal Sangar, actually, I don't have a very straightforward answer for that. It's just that the major impetus has always been towards crop management and within the crop management. In storage pest management, the options are always being looked at towards storage godowns, etc. But that's a good question. Actually, I should ask my country team and pest management team. But I know we all have products from industry which are useful, which can be used. Thank you for that question. Sorry, I know I didn't answer your question, but still.
Yeah.
Thank
Okay.
you.
Dr. Rahman, please come in.
Yeah, sir. First instance, it is an excellent presentation, sir. And also it is an excellent opportunity for me to see a lot of my guru age colleagues, Dr. Ramesh Babu, Dr. Abraham Varghese, and Dr. Varaprasad. Thank you very much. Now, couple of queries, Dr. Srinivas, after an excellent presentation. I was just trying to see that is there any opportunity in some of the technologies what you talked about. In fact, with regard to that sterile insect technique, which is being successfully used by Oxitec in Brazil. Then also, I remember in case of our ICMR also, Indian Council of Medical Research, I am authorized to talk about it because by being there in that project as a committee member, expert committee member. In fact, Oxitec has also presented along with Mahiko Monsanto for the approval of this one with ICMR. Somehow, because ICMR was very scary about the regulatory mechanism, which I don't want to go much in detail because by being a GAC member, I should not talk about it much here. But the other project which was approved and which is already successfully now, it is third or fourth year, wherein I need to put my input and how that can be applicable for the agriculture use. Like in case of Aedes aegypti, Wolbachia mediated mosquitoes. In fact, the mosquitoes has been sent from our country to Monash University in Australia. They have put their strain of Wolbachia into it. Then it came back to ICMR, and I think it is 8th or 9th generation that is being successfully reared in VCRC, Puducherry, Vector Control Research Center. My point of thing is, one of the greatest disadvantage, why for field release for last 3 years we are hesitating because of the public uproar. Why? Because you have used the very word, friendly insect. Friendly insect is the maybe acceptable word. Friendly mosquitoes we wanted to talk. So one of the control measure which we wanted to do is that instead of going for traditionally like fogging the colonies, we have to go and take mosquitoes and release.
Rahman, question please.
So that is the one where this can be useful for agriculture pests. Because in case of medical entomology, we face a lot of problem with regard to the public acceptance, releasing of the friendly insects. With regard to the GM studies, are there any, other than the example what you have given, are there any other successful example where this SIT is used by genetic manipulation? That is one question.
Yeah.
Other very quick question is, I will complete it, that with regard to the fall armyworm also, some of the proposals has already come So what is the status of the proposal? Probably you'll be the right person to tell. These are two my queries. One is whether agriculture, SIT, are there any examples which are already there in the field? Thank you.
Yeah. To quickly answer, sir, I think I spoke about agriculture pests, and one of the well-studied one is Falormium in Brazil, and it's not studied outside. It's studied in a controlled conditions, and the results were found to be very good. With respect to India, I have zero knowledge on what is being brought in agriculture pests, but I do, and I'm aware of Dr. Tyagi and it's GBIT actually from India who are working with the Oxitect. And so there's a lot of value in agriculture, but of course, you know better than me the regulatory system, how it's functioning and how fast we can bring this into the country. So yes, sir. That's my response basically. I'm not aware of any agriculture pests in India as of now.
Okay. So there is a query from Mr. M.H. Kodan Ram.
Yes,
Good doctor, should we work on pest-specific IRM strategy in high pesticide input crops instead of high pesticide input crops instead of simply recommending a rotation of insecticides?
I completely agree with you, sir. Pest-specific IRM strategy, or I would say a little crop-specific, because there are multiple pests in some high-input crops. So to look at the pest systems and of course, rotation of insecticides will be there if you are having so many or more than one or two target pests in a crop. So it's an integral part.
Okay. Any one or two questions? Last couple of questions. Anybody wants to come in? I think we have done a wonderful question answer session also.
Yes.
Let us take the input from Varghese.
Varghese from Bangalore.
Dr. Varghese, yeah, I called for him. Yeah, Dr. Varghese, are you there?
I am there. Can I give some few quick comments?
Okay, Dr. Varghese, I think, yeah, please.
At the outset, Dr. Srinivas, congratulations, excellent talk.
Thank you.
I wish your mother very speedy recovery. And I want a non-technical comment, the style of presentation, the pace with which you communicated is excellent. It's something which good slot, good words, good communication. Thank you so much. I have some.
Okay, sir, please be a bit louder, sir, because your voice is not that loud.
Okay, I have some suggestions. Can you hear me?
Yes, sir. Yes, sir.
See, some suggestions. For example, let me put off my video. You can hear better. I have some quick suggestions, Dr. Srinivas.
Yes, sir.
I would personally like if Bayers can come out with some kind of field kit where we can quickly identify in a particular pest, what is the group, which mode of action is resistant to, which it is not, So because there's an area of 30 kinds of actions mentioned, but we need to have a kit also parallelly, and only an organization like yours, would be able to come out with. That's the only suggestion I have at this point of time. Thank you, sir.
In fact, I didn't mention it very explicitly, sir. Bayer kind of has a complete set of teams that works on this area of mode of action determination and which chemistry goes into which mode of action. It's all done at the discovery level and testing level even before the product comes out. In relation to even a pest, so it is done. I just did not very explicitly mention it. We have a big team in Germany that works on, and it is substantiated by field level data that is collected in the countries where the product is set for commercial launches. Yes,
okay.
Sir. But it's a very valid.
Thank you.
Yes, sir.
Thank you.
Okay, thank you, Dr. Varghese. I think there is one quick comment from someone. The name is not very clear. frequent occurrence of pink bollworm in cotton growing regions. What further strategies need to be worked out? Do you have something to say?
I beats my knowledge, sir, right now. The way pink bollworm infestations are. I don't have a perfect answer except chemistry interventions that have been used. And of course, if somebody can bring in a technology which is more like GM insect based, if Oxitec has some, that will be one opportunity.
There is one, I think, last question from Director CPMB. After Varghese, I have a question. Dr. Mohan Kumar, please go ahead, sir. Dr. Mohan Kumar, please go ahead. Dr. Mohan Kumar, are you there? Director CPMB. Dr. Mohan Kumar. Okay.
Thank you, sir.
Okay. Any more questions from anybody from the audience? Okay. If there are no questions, Dr. Parimi, again, I would like to congratulate you on behalf of Plant Protection Association of India. And I wish your mother a speedy recovery. And we look forward to seeing more of, industry participation. And this is really enlightening on the, new strategies in IRM and pest management, new pest management strategies. And I request Dr. Srinivas to give me a write-up on this presentation. And we would like to bring out a special issue on these series of lectures. And I look forward to Srinivas to help me get in more, international speakers on various subjects. I would like to bring in someone. I'm looking for someone on artificial intelligence and machine tools in crop health management. Also, I'm going to bring in one expert on in the context of plant health management, the hydroponics, and many other areas of our interest. There are many people expressing interest in these areas. Also, we had a talk on space entomology and many such wonderful presentations. I look forward to Dr. Srinivas to give me a write-up as soon as possible.
Yes,
and
sir.
again, I wish your mother a speedy recovery.
Thank you, sir.
And now I request Dr. Saravanan to present a formal vote of thanks.
So thank you, sir.
Yeah, please go
So
ahead.
on behalf of Plant Protection Association of India, with pleasure, I formally propose vote of thanks. First of all, I thank Dr. B. Sarath Babu, sir, President of PPAI, for his constant motivation and encouragement and support to organize such a Golden Jubilee lecture series. And this is the 5th of its one. And it is his initiative to identify the topic and the speaker. And we really thank you, sir, for all your support. And also profusely thank Dr. Srinivas Parimi, sir, as a regional resistance management lead, Asia Pacific, Bayer Crop Science, Singapore, for readily accepting our invitation and sparing his valuable time in spite of his family problem. So thank you, sir, for sparing your valuable time and giving a very excellent and very informative and interesting topic. And they have covered, highlighted different approaches, tools, and the latest and emerging technologies, especially for managing insect resistance. It's a really eye-opening talk, sir. So our participants have already commented a lot, so I don't want to say much. So thank you, sir, once again, and also pray that mother will recover speedily. And I also thank our vice presidents of this society, Dr. Celia Chellam, head division of plant quarantine, and Dr. Jagdishwar, direct research, PGTSU, Hyderabad, and Dr. Srinivas Prasad, head plant pathology, Indian Institute of Rice Research, Hyderabad, for their constant support and encouragement in organizing such webinars. And also, I thank Dr. Anita Kodaru, Officer in Charge, NPPG Regional Station Hyderabad, for all her support for conducting this webinar and also providing necessary facilities for this seminar. And also, I thank executive members of Plant Protection Association of India, Dr. Parameshwari, General Secretary, Dr. Bhaskar, Treasurer, and Dr. Kavita Gupta and Prasanna, Associate Editors, and all the councillors representing from various zones. So also, I especially thank our distinguished guest today, Dr. Varaprasad sir, and he is always our inspiration, and he will be always with us in inspiring us. And also, I thank Dr. Abraham Varghese for his valuable comments, and his presence really makes a difference. And I also thank Dr. Ramesh Babugaru for giving his valuable comments and remarks, and for Dr. Raghuman sir for his valuable comments and also the questions, and also all other participants who have given valuable suggestions as well as the post questions. And we feel that we understand that this seminar will be a fruitful one. And above all, I thank each and every participant who actively participated and posed questions. And I understand that it's a valuable time that you have spent, and still it shows that you are really hunger for knowing the knowledge and spread and manage this insect problem which are affecting our food security. So if at all I forget anyone to mention by name, please pardon me, and hoping forward to see you more people in the coming series. Thank you once again.
Thank you, sir.
Thank you, Sini.
Dr. Srinivas, before we close, I think my audio was muted. Now I think I am able to, you are able to hear me. You know, Dr. Mohan Kumar has come back with his a small little query: ginning industry role in PBW management. Dr. Srinivas, are you there?
Sir, he told that he will catch up offline. Thank you so much.
Okay. Thank you very much. Thank you so
Thank
much.
you.
Thank you all once again.
Thank you all once again, Dr. Raghuman sir, Dr. Ramesh, and all others. Thank
Dr.
you very
Mohan.
much, sir.
Take care, sir.